So we're going to take some poop bacteria and put it in that bioengineered human intestine and see what happens. What a time to be alive! If you walk into an empty room, you might think you're alone, but in fact you are hanging out with trillions of bacteria that live on and in your body. There are more bacterial cells in your body than your own cells. We call this community of bacteria our microbiome, but don't freak out. Our bacterial friends are a normal part of our lives. In fact, microbiomes are a super hot topic in science right now because we're discovering that they play a major role in maintaining our health and well-being. On that note, there's someone I want you to meet. This is our super special guest, Dr. Jemila Kester. She's a postdoctoral scientist in the Griffith Lab in the bioengineering department at MIT, where she studies the microbiome of the human intestine, aka the gut.
Yes, and we specify that it's the gut microbiome because each part of your body has different bacterial communities. You won't find the same bacteria in your mouth, your nose, or on your skin as you would find in your gut.
I sure do hope not.
In your gut, there's several different kinds of bacteria, including E. coli, the good kind, not the bad kind. When your gut microbiome is happy and functioning, it helps your digestive system work, and it also prevents disease-causing bacteria from establishing in your gut.
So how does our gut microbiome become unhappy?
Okay, let me start by saying that antibiotics are some of the most important drugs ever discovered and have saved countless lives. So if your doctor prescribes them to you, you should take them. But an unintended consequence of taking antibiotics is that they kill some of the good bacteria in your body, which are there to prevent bad bacteria from growing. Most of the time, taking antibiotics just gives you some minor stomach upsets, but in extreme cases, a really bad bacteria called C. diff can colonize your gut and make you extremely sick. Conventional treatment uses super specific antibiotics to kill the C. diff, but sometimes that doesn't work and we want to fix the microbiome. And to do that, we use something called a fecal transplant.
Is that what I think it is?
Yes. A fecal transplant is healthy poop that is a pill that you swallow or is a slurry that's put up your butt. You take poop from a healthy donor and you put it in a sick person to make their microbiome better. We know that fecal transplants are over 90% effective at treating otherwise incurable C. diff, but we don't really know what the fecal transplant is actually doing. So what bacteria are actually working? We don't know. And wouldn't it be great if we knew which bacteria cause people to get healthy again, and then we could transplant just those instead of poop from a rando person?
Yes, less rando poop sounds ideal.
So that's the experiment we're going to run today. We want to know how the cells in our gut react to fecal transplants with different microbial communities. Maybe some combinations of bacteria are better for our guts than others. Let's get started.
Oh, no, shut it down, shut it down.
Oh, no. We wouldn't do this type of experiment on a human subject.
Oh, thank goodness.
This is a bioengineering department, so we'll be doing this experiment on a bioengineered synthetic human intestine. So this is the gut on a chip. It is a bioengineered synthetic human intestine that allows us to do microbiome studies on human cells.
What? This is an intestine? How?
We've designed it to mimic the physiological aspects of a gut. In each chamber, we're growing living human intestinal cells on a membrane, and then this represents the wall of the intestine. The space above the layer of cells represents the lumen, which is the hollow tube of your gut. The cells are bathed in a fluid that matches what you'd find in your own intestine. It's got the right pH, salts, and nutrients. The lumen of our gut is anaerobic, meaning there's no oxygen in there at all. We keep the lumen of our gut on a chip anaerobic too, by pumping in nitrogen gas. So in a real intestine, the tissue below the intestinal wall will be exposed to oxygen because it has blood vessels which have oxygenated blood. This results in a gradient of oxygen from none in the lumen to lots in the deeper tissue. We have that same gradient in our system, so the environment underneath the cell layer is exposed to oxygen.
Wow, so you have this whole intestinal ecosystem that you can manipulate for your experiment.
Yeah, so now we can add different things to our system, like bacterial cells from a healthy donor or antibiotics, and see how they interact with the gut cells and each other. So for this experiment, we want to find out if certain bacteria that might be found in a fecal transplant are better for the gut than others. We'll be working with two common gut microbiome bacteria, E. coli and B. theta. We'll test how the gut cells react to each of them separately and in combination. First, we have to grow the bacteria. Now, they've been isolated from human fecal samples.
How do you grow bacteria in the lab without oxygen? Are you being facetious?
We use this special anaerobic chamber. It's completely sealed off from the outside environment, and we pump in nitrogen gas, which pushes all the oxygen out. This is the liquid media. We'll grow our bacteria in here. It has lots of yummy food for the bacteria to eat. This is the plate that has our bacterial culture. We'll use this to inoculate the media. And now we'll put them in this incubator to grow overnight. Yay! The bacteria are ready.
And now it's time to add them to the gut on a chip?
Yep.
Just so you know, we would normally do this part of the experiment in something called a biosafety cabinet. It's basically a bacteria-free zone that would keep the gut on a chip from getting contaminated with bacteria in the air. But they are super loud and really annoying to film in, so we're doing this out in the open instead.
So now we add the different bacteria combinations to the chambers. Each chamber is self-contained, so we can test each combo separately. Then we put the gut on a chip in an incubator, so it stays nice and warm and moist for 7 days.
And now they're ready. So how do we tell which bacterial cocktail is best for the gut cells?
So this is still ongoing work in the lab. In fact, one of my undergrads is working on this project even as we speak. But I can tell you what the plan is. We know that fecal transplants help our guts to produce more antimicrobial peptides. They are molecules that are part of a good immune response, and they help our bodies to kick out bad bacteria like C. diff by targeting them for destruction.
Whoa, so the good bacteria cause our own cells to make those peptides?
Yeah, exactly. And so we can compare levels of gene expression of these peptides across the different combinations and see which bacteria produce the best immune response.
So just as a quick refresher, all of our cells contain the same set of genes, or DNA. Think of it as a big, beautiful genetic library. But not all cells express the same genes. They take different books out of the library. Like an intestinal cell does not need the same books as an eyeball cell. In order for a gene to be expressed, it needs to be copied into a slightly different molecule called RNA, and then translated into a protein which actually does a job in the cell. We can measure levels of RNA to figure out which genes are being expressed.
Right, so we'll extract RNA from the human cells, and then we'll measure the levels of expression of the antimicrobial peptides and hopefully figure out which combination of bacteria does the best job.
I can't wait to hear about your findings. What are the next steps of your experiment?
So my ultimate goal is to be able to tailor treatment for each patient, because not all humans will react the same way to the same microbial community. We'd like to be able to run this assay with a patient's own cells and different microbes to be able to see which treatment is actually best for them.
Wow. Jemila, thank you so much for taking the time to show us the wild and wonderful world of gut microbiome research.
You're welcome.
Don't forget to subscribe to Science in Real Life. If you want to know more about Jemila's research, you can check out the Griffith Lab website, and we'll see you next time.